Namaste students. We perform many activities consciously or unconsciously. When we eat, our eyes helps to locate the food. The nose smells it. Hands bring the food to the mouth, which masticates the food. Teeth chew and the tongue helps to move the food into the digestive system. All these activities need to happen in a coordinated manner. The organ system in our body that helps in coordination and integration of body activities. The nervous system involved as a means to sense and respond with the absolute precision to changing conditions inside and outside the body. The information has to be exchanged between cells distant from each other. Information about changes in the external environment has to be received and then transmitted to the cells situated far away in the body. For control and coordination of the body, two types of systems have been developed in animals. One is endocrine system and is the nervous system. Endocrine system you have already studied. Now we can study about the nervous system. Next, why we need the nervous system? Nervous system is the main coordinating and integrating system of body activities. Its major functions are: first one, keeps us informed. It gives the information about the outside world through the sense organs. With the help of the sense organs, we can get the many information. Through the eyes, we can see the things. Through the ear, we can listen the information. So like that, it keeps us informed. Next, enable us to remember, to think and to reason out. It helps to remember the things, thinking power as well as the reasoning. Next, controls and harmonizes all voluntary muscles activities. For example, running or even holding the book in our hand while we are reading it. While we are reading, we are holding our books. So it controls and harmonizes all voluntary muscular activities. Next, regulates involuntary activities such as breathing and heart, working of the heart. Without our thinking, we are doing these works. It regulates the involuntary activities. For these things, we need the nervous system. Next, structure of neuron or nerve cell. Neuron or nerve cell, it is the structural and functional unit of nervous system. Neuron or nerve cell is the structural and functional unit of nervous system. Our nervous system consists of brain, spinal cord, as well as sense receptors and a whole lot of nerves. First, we can study the structure of neuron. In the neuron, we can see three important parts: cell body, dendrites and axon. First, cell body, or it is also called perikaryon or cyton. Cell body has a large central nucleus surrounded by granular cytoplasm. In the cytoplasm, we can see nasal granules and neurofibrils. Along with this, other cell organelles also present like mitochondria, Golgi complex, endoplasmic reticulum, etc. But there is no centrosome. There is no centrosome in the cyton because the nerve cells have lost the ability to divide. A centrosome is absent in the nerve cell. That's a nerve cell lost the ability to divide. So this is the cell body or perikaryon. Next, dendrites or dendrites. Several short thread-like branches are called dendrites. They are arise from the cell body. The dendrites conduct nerve impulses to the cyton. Dendrites conduct nerve impulses to the cyton. These are nothing but cytoplasmic projection. Here the cytoplasm which are present in the cell body, they are projecting out, so it forms a thread-like structure that is called dendrites. It helps to transfer the nerve impulses to the cyton region. Third part, that is the axon. Axon is a long process from the cell body. It varies in size from a few millimeters to even more than 1 meter in length. In most neurons, the axon is surrounded by a white insulating sheath called myelin sheath or medullary sheath, which is covered by an outermost thin sheath called neurolemma. It has a two layer. Outermost layer is called neurolemma and the inner layer is called myelin sheath. It is act as a insulating. It avoid the mixing of nerve impulses. So it acts as a insulating sheath. The myelin sheath shows gaps throughout the length, which are called nodes of Ranvier. Myelin sheath is discontinuous sheath, so it has a in the middle we can see a gap. That gap is called node of Ranvier. But neurolemma, it is a continuous layer. Some axons may have side branches called collaterals. The end portion of the axons have swollen ends, that is called bulb-like structure. In this bulb-like structure, it has a chemicals called neurotransmitters. The name of the chemical is acetylcholine. The name of the neurotransmitter is acetylcholine. At the end portion of the axon, it has a swollen bulb-like structure. That in the bulb-like structure, we can see the chemicals called neurotransmitter. Example is acetylcholine. The axon terminals or axon ends are closely placed near the dendrites of another one or more neurons, but are not connected. Such gap between are called synaptic cleft. Here, between the one end of the axon of the neuron and the dendrites of another neuron, between these two there is a gap. That gap is called synaptic cleft. Here two neurons are not connected directly. Between this axon end of one neuron and dendrites of another neuron, between these two there is a fine gap is there. That gap is called synaptic cleft. Now you can see structure of neuron. It has a thread-like structure that is called dendrites. Dendrites are arrays from the dendron. Dendrons are nothing but it is a cytoplasmic projection of the cyton. And in the cyton part, it is called as perikaryon or cell body. We can see the nucleus and cytoplasm. In the cytoplasm, we can see the Nissl granules and other organelles also. One of the dendron, it become elongate, that is called axon. It is surrounded by myelin sheath as well as neurolemma. The myelin sheath is discontinuous sheath. The between that gap is called node of Ranvier. And axon end is branching, and at the end of the branch we can see a bulb-like structure. That in the bulb-like structure we can see a neurotransmitter. This is the structure of neuron. Next, transmission of the nerve impulse. In the normal, means resting condition, the outer side of the nerve fiber carries positive charge. This is called polarized state. This polarization is due to more sodium ions outside the axon membrane. If there is no stimulus in that situation, the more positive charges we can see outside the nerve fiber. that is in the outside the axon membrane, because it has more sodium ions. That state is called polarized state. On stimulation, it may be the mechanical, electrical or chemical or heat. On stimulation, the axon membrane at that spot becomes more permeable to sodium ions, which move inward and cause loss of polarization. That is called depolarization. And that state is called excited state or excited region. When the axon receives the stimulus, in that condition, whatever the sodium ions are there outside the axon membrane, it enters inside the axon membrane. Because in that condition, axon membrane become more permeable to sodium ion. So, sodium ion moves inward. When the sodium ions moves inward, the outer axon membrane, it loses its polarization. So, it turns into depolarization. And that depolarization state is called excited state or excited region. The point of depolarization becomes a stimulus for the next neighboring area of the membrane, which in turn becomes depolarized. So when it receives that sodium ion, when it moves inside, so it then enters the next region of the axon. It causes the next region as a depolarization. Meanwhile, the previous area becomes repolarized due to active transport of sodium ions again to the outside. This transport is achieved by what is called sodium pump, using energy through ATP. Here, when the depolarization takes place, that sodium ions, that impulse or that stimulus, it passes to the next region of the axon membrane. When it passes to the next region of axon membrane, that region will become depolarization or excited region. But the previous region become repolarized. Previous region become repolarized, means in the previous area of the axon membrane, the sodium ion which moves inward, it again moves outward direction. When it moves outward direction, that area become repolarized, and the next region of the axon membrane undergo the depolarization. So like that, the nerve impulse is transmitting. You can see in the diagram, the first one is the resting stage, means sodium ion is outside the axon membrane. So it is called as resting stage or polarized state. So when the axon membrane receives the stimulus, whatever the sodium ion is there outside the axon membrane, it enters inside. So that area is called excited region, and that is called depolarization. But the next part of the axon membrane is still in resting region. In the diagram B you can see. And in the diagram C you can see that whatever the sodium ion moves inside, it passes the stimulus to the next region of the axon membrane. When it passes the stimulus to the next region, in the previous region, the sodium ion moves one side outside the axon membrane. When it moves one side again outside the axon membrane, that is called recovery region or repolarization. It comes to the normal state. But the next region undergo the excited region. So through this, nerve impulse is transmitting. Next, synapse. Synapse is the point of contact between the terminal branches of the axon of a neuron with the dendrites of another neuron separated by a fine gap is called synapse. Synapse is nothing but it is a point of contact between the axon end of one neuron and dendrites of another neuron, and both are separated by a synaptic cleft or fine gap, that is called synaptic cleft. So that is called as a synapse. The gap is called synaptic cleft, the junction of the contact between the two neurons, that is a axon end of one neuron and dendrites of another neuron, including synaptic cleft, is called synapse. At a synapse, the nerve impulse jumps into the next neuron. In the synapse region, nerve impulses jumps from axon end of one neuron to the dendrites of another neuron. This is a chemical process because it takes place with the help of the chemicals. As the impulse reaches the terminal end of an axon, a chemical called acetylcholine, it acts as a neurotransmitter, it is released. This chemical sets a new impulse in the dendrites of the adjacent or next neuron. This acetylcholine helps the transmission of nerve impulses from axon end of one neuron to the dendrites of another neuron. After passing the impulse, this chemical is soon broken down by an enzyme to make the synapse ready for the next transmission. After transmission of one nerve impulse, this acetylcholine breaks down with the help of the enzyme, and it keeps that synapse ready to receive the another new transmission. So this is called as a synapse.
A neuron transmits nerve impulses through itself, but when it's time to send a message to another cell, most neurons will send chemicals to the next neuron across a very small gap between them. The junctions between neurons and the next cell are called synapses. There are three major types of cells that neurons will form synapses with: sensory receptor cells, other neurons, and effector cells. A sensory cell could be part of the eye, ear, nose, skin, or any place where senses are used. This sensory cell is connected to the skin and will pick up pain sensations. The pain message may be sent to another neuron that could be part of the spinal cord or brain. These intermediate neurons are also called interneurons. The message can then be sent to a cell that will have an effect on another cell, so they're called effector cells. In this example, the effector cell is going to signal a muscle cell, which would move this area of skin away from the source of pain. These three cell types together make a simple reflex arc, going from sensation to the central nervous system to an action. The chemicals released into the synapse are called neurotransmitters. Many neurons have a very small fluid-filled gap between the presynaptic cell and the postsynaptic cell. This gap is called the synaptic cleft, and it's only about 20 nanometers wide. It's a really, really tiny gap. Now that we know some of the basics, we can look at the steps of synaptic transmission in more detail. First, a nerve impulse in the presynaptic neuron reaches the end of the membrane. This causes calcium ions to diffuse into the cell through channels in the membrane. The influx of calcium causes vesicles with neurotransmitters in them to move and fuse to the cell membrane. The neurotransmitters are released into the synaptic cleft by exocytosis. They diffuse across the synaptic cleft and bind to receptors on the postsynaptic membrane. The binding of neurotransmitters triggers sodium ion channels to open and pass the threshold potential. The action potential is propagated along the postsynaptic neuron. Then the neurotransmitters are broken down and removed from the synaptic cleft. If there isn't enough neurotransmitter binding to receptors, then there won't be enough sodium flowing into the cell to reach threshold potential. This means that there is an all or nothing response. Either there is enough neurotransmitter to start the action potential and the impulse is sent, or there is no impulse. In this case, there was not enough neurotransmitter released to trigger an action potential, so the neurotransmitters will be broken down and the sodium-potassium pumps will restore resting potential in the postsynaptic cell. There are many different types of neurotransmitter, but we'll look at just one in more detail right now, acetylcholine. This neurotransmitter is made of an acetyl group and a choline, which must be combined together in the presynaptic cell before it's packaged into a vesicle. Then it can be released into the synaptic cleft. After acetylcholine binds with a receptor, it is rapidly broken down by acetylcholinesterase, an enzyme that's present in the synaptic cleft. The acetyl group and choline can then be reabsorbed by the presynaptic neuron and combined again into acetylcholine for reuse. One type of insecticide takes advantage of the acetylcholine receptors to paralyze and kill insects. Neonicotinoids are synthetic compounds that are similar to nicotine but bind permanently to the acetylcholine receptors. The acetylcholinesterase cannot break the neonicotinoids down, and the neurons can no longer receive messages or send action potentials. This is what leads to the paralysis and death of the insects. The effects are very small in humans and mammals, but the neonicotinoids don't discriminate between types of insects. Honeybees and other beneficial insects are inadvertently killed by neonicotinoids too.
Next, types of neurons. There are 3 types of neurons we can see. First one is sensory neurons. Sensory neurons convey the impulse from the receptor, means sense organs, to the main nervous system, that is the brain and spinal cord. Sensory neurons, it carries the impulses from sense organs and send that impulses to the main nervous system, that is to the brain or spinal cord. Next, motor neurons. It carries the impulse from the main nervous system to an effector. Effector includes muscles or glands. Motor neurons, it convey the impulses from brain or spinal cord to the muscles or glands, or it convey the impulses from the main nervous system to an effector. Next, association or connecting neurons. It is located in the brain and the spinal cord, which interconnect the sensory and motor neurons. Association neuron, it connects the sensory neurons as well as the motor neurons, and we can see it in brain and spinal cord region. Next, nerves. Nerve is a bundle of nerve fiber, that is axons of separate neuron enclosed in a tubular sheath. Nerve is nothing but it is a bundle of nerve fibers, means it is a bundle of axons of separate neurons. It is enclosed by a tubular sheath that is called as a nerves. Nerves are the thread-like white structure which emerge from the brain and spinal cord and branch out to almost all parts of the body. A nerve may be compared to an underground electric cable containing numerous conducting wires, each insulated from the other. Here, the myelin sheath of the axon acts like an insulation and prevents mixing of impulses in the adjacent axons. So, myelin sheath acts as an insulating sheath because it avoid the or it prevent the mixing of impulses in the adjacent axon. Types of nerves. In the nerves, we can see the 3 kinds of nerves. First one is sensory nerves. It contains only sense fibers, bringing impulse from the receptor, that is sense organs, to the brain or spinal cord. Sensory nerves contain the sensory fibers. It carries the impulses from the receptor, that is sense organs, and it give that information or impulses to the brain or spinal cord. Example, optic nerve arising from the eye and ending the brain. Optic nerve, it is a nerve, it carries the impulses from eye to the brain region. Next, motor nerve. It contains only motor fibers carrying the impulses from the brain or spinal cord to effector organs to bring them into action. Here, motor nerves, it contains a motor fibers. It carries the impulse from brain or spinal cord and send that impulse to the effector, that is the muscles or glands. Example, a nerve arising from the brain and supplying the muscles of the eyeball for rotating the eye. Next, mixed nerve. Mixed nerves are those that contain both sensory and motor fiber. Mixed nerve contains both sensory as well as the motor fiber. Example, a spinal nerve. Spinal nerve act as a mixed nerves. You have to know some of the basic terms. First one is stimulus. Stimulus is an agent or a sudden change of the external or internal environment that results in a change in an organism or any of its body parts. That is called stimulus. Stimulus means which brings changes in our body, inside or outside the body, that is called as a stimulus. Next, a response. The change in an organism resulting due to stimulus. So what changes takes place in the organism body because of the stimulus, that is called response. Next, impulse. A wave of irritability means electrical disturbance that sweeps over the nerve cell. Next, receptors. The specialized epithelial cells which on receiving the stimulus set up waves of impulses towards the central nervous system. Receptors means which receive the stimulus, that is called as a receptors, and that receptors it sends this stimulus in a wave of impulses towards the central nervous system. Effectors. Muscles or glands which on receiving the impulse from the brain or spinal cord contract or secrete substances. Here muscles or glands act as a effector. It receives the impulses from the central nervous system, that is a brain or spinal cord, and when it receives the impulse in the muscles it shows the contraction, and in the gland it secretes the chemicals. Here receptors are our sense organs. Effectors are muscles or glands which are present in our body. And next one is the ganglia. Ganglia are the aggregates of the nerve cells, it means cell bodies, from which the nerve fibers may arise or enter into. Here it is a aggregate of the nerve cells, especially the cell body, from which the nerve fibers may arise or enter. That is called as a ganglia. In today's class, you have studied about the structure of the neuron, types of neuron, as well as the nerve also. And still more information is there in the next class. And this chapter is very important. And structure of neuron is also very important.